my name is Rachel Recton, and I will be talking to you on the topic of neurotransmission. So we're going to go over this graphic in just a little bit. I'm going to kind of let you know the different definitions of each of these. It's just the parts of the neuron, what it is. We have the dendrite, which receives the information from the terminal buttons of other neurons and helps pass this information to other neurons. Soma is the cell body of the neuron. The axon is a long, thin, cylindrical structure that conveys information from the soma of a neuron to its terminal buttons. And that kind of goes into the next one, which is the myelin sheath, the sheath that surrounds the axons and insulates them from preventing messages from spreading between adjacent axons. And this myelin sheath kind of all this just ties together as you go through the part of the neuron. Next is the node of Ranvier. It's a naked portion of the myelinated axon between adjacent Schwann cells. And then next kind of goes into the Schwann cell, which is in the middle of kind of like the axon with the myelin sheath. It's in the peripheral nervous system that is wrapped around the myelinated axon, providing one segment of its myelin sheath. And lastly, the axon terminal button is the bud at the end of the branch of an axon, forms synapses with other neurons, sends information to that neuron. So just going back real quick to show you, if it will let me. So let's see, previous. Here we go, there we go. So here we have those tree-like structures, the dendrite. The soma is the cellular structure of the body. Axon right here, that's the cylindrical with the myelinated sheath. Right here is those Schwann cells with that one little sheath of theirs kind of peeking out. The node of Ranvier is that naked part of the axon where the myelinated sheath does not cover it, and then the axon terminal right there, those are the buds. So going back through, this kind of leads up into talking about what the action potential is. The membrane potential is negatively charged. The type of electrical charge that draws a line across the membrane is called the resting membrane potential, and that is caused by many electrodes that you can use with this oscillator. Put it inside one of the neurons, and it will show you that the resting membrane potential is actually at 70 millivolts, but since it is negatively charged, it would be negative 70 millivolts. Adding a positive charge through the membrane can cause the axon to depolarize. Just because since it is negative, adding a positive charge to it can cause the membrane potential to diminish. After depolarization, the membrane potential will suddenly try to reverse itself. And the way to do that is to go back to normal, it must hyperpolarize. And what that does is that normally if it was negative on the inside and positive on the outside, it tries, with that positive charge going through it, it makes it positive, makes the outside negative. The rapid reversal of the membrane potential is called the action potential. Diffusion and electrostatic pressure, what that is, the diffusion is the movement of molecules from regions of high concentration to regions of low concentration. The diffusion process causes something that's electrically charged ions. There's two types of ions. There's cations, which is the positive, and there's anions, which is the negative. The electrostatic pressure is the force exerted by the attraction of the opposing ions or the repulsion of ions that are the same charge. What that is, so say there is a cation and an anion, having those two together, they will attract, rather than if I had two anions together, it would not attract, the force would repel from each other. Right here we kind of have the diffusion kind of process right here. It kind of talks about with the 4 different types of major ions, which is potassium. There's organic anions. Organic anions can only be in the intracellular portion. We have the cell membrane right here. And then here we have the extracellular fluid as well. And here we have the sodium, the chloride, and the potassium. And then this is just kind of explaining, this is a sodium-potassium pump, explaining how it works when you add in the 3 sodium and out will come out to potassium. The action potential has an all or none law in which it's either the action potential is there or it is not. There's no way to kind of stop it. Action potential can be split in the sense of once it hits one of those dendrite branches, it can be split, but it cannot grow or diminish in size. And then the action potential variable is represented by an axon's rate of firing. And that's just kind of measuring the different types of action. Going on, we have the chemical components of the action potential. We have the synaptic transmission, which is the transmission of messages from one neuron to another. Going on, we have the postsynaptic potentials. Alterations in the membrane potential of a postsynaptic neuron produced by release of neurotransmitter at the synapse. We have the binding site, in which that's the location where a receptor protein to which a ligand bonds, and a ligand is just one of those chemicals that's able to, as going on to the next one, it's a chemical that binds to the binding site receptor. In order for this to go, once the ligand is binded with the binding site, what that does is help let ions either be passed or not be able to pass depending on the charge. Here we have the synapse graphic, in which we have the kind of postsynaptic down here. Here we have the synaptic cleft. And what this is right here is that whenever the synaptic cleft binds together, it kind of, or once the, I'm losing it. The postsynaptic receptors open neurotransmitters. Going on, here we have the neurofilaments, the vesicles, in which that is just little filaments inside the binding site here. And going on, so there are three types of sites for synapses. There's on the dendrites, on the soma, and other axons. The presynaptic membrane is the membrane of a terminal button that lies adjacent to the postsynaptic membrane and through which the neurotransmitter is released. Postsynaptic membrane, the cell membrane opposite of the terminal button in a synapse, the membrane of the cell that receives the message. And then the synaptic cleft is the space between the presynaptic membrane and the postsynaptic. So like in the previous graphic I kind of showed you, it has the synapse kind of right here, and then the postsynaptic membrane. So you have the presynaptic and the postsynaptic, this little area right here that was the synaptic cleft. The synaptic vesicles, a small hollow bead-like structure found in terminal buttons, contains molecules of a neurotransmitter. The action potential is sent down in axons. Inside the terminal buttons are several synaptic vesicles fusing with the postsynaptic membrane and then breaking open, kind of releasing all of its contents within the synaptic cleft. The postsynaptic receptors, receptor molecule in the postsynaptic membrane of a synapse that contains a binding site for a neurotransmitter. Binding occurs, the postsynaptic receptor opens neurotransmitter-dependent ion channels, and the direct ion channel, when a molecule of the appropriate neurotransmitter attaches to the binding site, the ion channel opens. In the indirect ion channel, some receptors do not open the ion channels directly. It instead starts a chain of chemical events. And what that is, it has the G protein, a protein that is adjacent to a metabotropic receptor. Conveys messages to other molecules with a ligand bind and activate the receptor. Second message is a chemical produced when a G protein activates an enzyme, carries a signal that results in opening of the ion channel or causes the event to occur in the cell. And that kind of that's the end of our presentation.